Welcome to this explainer. Consider this your definitive masterclass specifically designed to help you secure your highest possible marks in the grade 12 Life Sciences mid-year exams. We're treating this upcoming exam not just as another test, but as a critical milestone for your final National Senior Certificate. So we're using the 2021 examination guidelines as our ultimate strategic blueprint today. We're going to decode the absolute trickiest processes from term 1 and 2, think DNA, genetics, and responding to the environment. We'll highlight exactly where the bulk of your marks actually come from, and we'll expose those technical red zone pitfalls that literally cost students top grades every single year. Let's get into it.
Here's our blueprint for today. 1. Scope and exam strategy. 2. Decoding protein synthesis. 3. Meiosis and non-disjunction. 4. Human reproductive cycles. And 5. Essential exam instructions.
Okay, Section 1, Scope and Exam Strategy. Let's kick things off with a pretty sobering reality check directly from the 2023 Diagnostic Report.
So the absolutely crucial point here is that grade 12 life sciences does not exist in a vacuum. It's a 3-year qualification. That means topics from grades 10 and 11, like homeostasis, cell structure, photosynthesis, and population ecology, are entirely fair game. They can, and honestly, they will, be examined within the context of your grade 12 curriculum. You simply can't afford to leave that foundation behind.
Now, knowing exactly where your marks come from, that is strategic. Just look at the massive 40% weighting on responding to the environment and 27% on human reproduction in Paper 1. Wow, right? Meiosis might only sit at 7%, but it's completely foundational. Over in Paper 2, the human endocrine system is a heavy hitter at 15%. If you're figuring out how to manage your study time, use this breakdown as your compass. Focus your heaviest efforts on these core topics. And during the actual exam, don't burn all your time on low-weight introductory questions at the expense of these high-mark sections. You've got to maximize your return on investment. Moving on to Section 2, Decoding Protein Synthesis. Let's take a deep dive into this incredibly complex biological process. Protein synthesis is rigidly sequential, and you've got to treat it exactly that way in your exam answers. First up is transcription. A section of the DNA double helix unwinds and unzips, serving as a template. Then, free RNA nucleotides arrange themselves to form a complementary mRNA strand. Next comes the crucial physical transition. That mRNA actually leaves the nucleus, heads out into the cytoplasm, and attaches to a ribosome. Only then does translation begin, where transfer RNA, or tRNA, brings in specific amino acids to build the final protein. You have to use these precise definitions in your exam. Please do not mix up the synthesis of mRNA from the DNA template, which, remember, is transcription, with translation, which is the actual conversion of that mRNA code into a correct sequence of amino acids to form a particular protein. Precision here is totally non-negotiable. And this brings us to the rule of lethal spelling. Misspelling words like writing glucagon instead of glycogen or centriole instead of centromere literally changes the biological meaning entirely, and it will get you 0 marks. Accuracy is absolutely everything. When you're visualizing mRNA codons, remember they sit in distinct groups of 3 adjacent nitrogenous bases. Examiners love to ask you to use backward reasoning here. So, if they give you a tRNA anticodon, you must be able to work backward to find the complementary mRNA codon, and then backward one more time to find the original DNA triplet. It's exactly like solving a puzzle. Just remember, adenine pairs with uracil in RNA, and cytosine always pairs with guanine. All right, Section 3, Meiosis and non-disjunction. Let's examine exactly how cells divide to form gametes and the catastrophic consequences when these mechanics fail. The syllabus explicitly demands that you know the similarities and differences between meiosis and mitosis. The core difference? Mitosis maintains the chromosome number. Specifically, you have to know the difference between metaphase 1 of meiosis, where chromosomes align in homologous pairs at the equator, versus mitosis, where they align in a single-file line. Meiosis divides twice to produce gametes, and critically, it halves the chromosome number to form haploid cells, each with a slightly different genetic makeup thanks to crossing over. And here is the exact chronological failure that causes Down syndrome. This is the perfect spot to apply what we call the marking hierarchy. If a question just says name, you can simply state non-disjunction and move on. But for a 3-mark explain question, you must provide the full cause and effect timeline. It starts during anaphase, when homologous chromosomes or sister chromatids fail to separate. If this happens at chromosome pair 21, it leads to an abnormal gamete with 24 chromosomes instead of 23. When that abnormal gamete fuses with a normal one, the resulting zygote has 47 chromosomes. Writing out that specific sequence, that's what earns you maximum marks. Let's keep moving. Section 4. Human reproductive cycles. This endocrine flowchart is your absolute ultimate tool for cause and effect reasoning. Notice the pituitary gland at the top secreting FSH, which stimulates the follicle in the ovary. The developing follicle then secretes estrogen, which causes the endometrium to thicken. Later on, LH causes ovulation and the formation of the corpus luteum, which then secretes progesterone to further thicken that lining. So if you get a question asking you to explain a hormonal change, literally just follow these arrows. Every single action is a direct trigger for the next one. To really conquer those tricky graphical questions, you have to perfectly tie these hormonal secretions to their specific day ranges. Days 1 to 7 is menstruation, but simultaneously new follicles are already developing. Days 8 to 13 is the domain of estrogen, thickening the lining. And from days 15 to 22, progesterone steps in to make that endometrium even thicker, more glandular, and highly vascular to prepare for a fertilized ovum. Just like the negative feedback loops you study in glucose and osmoregulation, these hormonal shifts are a delicate, continuous cycle of triggers and responses. You need to know these day brackets like the back of your hand. Finally, Section 5, Essential Exam Instructions. Welcome to the red zone. Please do not let technical pitfalls ruin all your hard work. These are the exact instructions from the actual exam cover page. Start each question at the top of a new page. Do all your drawings strictly in pencil and label them in blue or black ink. This level of technical perfection applies across your entire paper. Remember the mark first rule: if an examiner asks for 2 reasons and you give them 4, only the first 2 are marked. Period. For investigations, precision is key. You've got to know the difference between reliability, which requires repeating the experiment or increasing your sample size, and validity, which requires controlling your constant variables. And for that perfect 6-mark graph, always explicitly identify your independent variable on the x-axis, your dependent variable on the y-axis, ensure a clear caption without ever using the word versus, and never forget your units. Failing to follow these basic directives results in unnecessary lost marks before they even start grading your biology. So, the authoritative blueprint is in your hands. We've mapped out the heaviest weightings, we've demystified the complex biological mechanics, and we've locked in the technical rules. The ultimate separator between an average student and a top achiever isn't just knowledge, it's precision and it's strategy. When you finally sit down with that paper, will you let technicalities steal your grade, or will you execute flawlessly? The choice is entirely yours. Thank you for joining this explainer, and I wish you absolute success in your exams.